Sweet. All right, folks. We're good to go with our next presentation. This one's got a bit of a bite to it, it was Deborah Hofstra that's doing it, but she's not here, so Daniel Clements is subbed in. Now, he's all the way from South Australia, went to get away from because the Wallabies weren't winning, and that's the rugby team, not the critter. But yeah, Daniel's going to talk to us about alligator weed genetics and influencing weed control outcomes. Daniel.
Thank you very much. Thanks for that introduction, very kind. So I'm presenting as part of Earth Sciences New Zealand, which is the merger of both NIWA and GNS that's recently occurred. Today I'm going to be talking about does genetic variation of alligator weed influence control outcomes. So I'm presenting this work on behalf of my colleague at Earth Sciences New Zealand, Deborah Hofstra, but as well as our collaborators at Mississippi State University. So I just thought I'd give a bit of a quick rundown. We've heard a fair bit about alligator weed already today and the issues that it's causing, but I'll give a rundown of alligator weed, the issues that it's causing, different weed control outcomes that have occurred from experimental studies in the USA and New Zealand, and then some more recent work that we've been doing on examining the genetic diversity of alligator weed populations in New Zealand, Australia, as well as some of the work off the back of some of the work that was being done in the USA. So alligator weed is quite unique. It's an aggressive invader of both aquatic and terrestrial environments. Introduced to Northland in ballast, discarded from ships in around 1906, so around the Dargaville area. It's now well established in Auckland, Northland, and the Waikato, particularly in the lower reaches of the Waikato catchment. Scattered infestations in the Black Bay of Plenty, infestations in Manawatu, Wanganui, around here, Palmerston North, Taranaki, Hawke's Bay, Wellington regions have also had outlier infestations as well. It does pose a significant threat to New Zealand's waterways, wetlands, floodplains, irrigation systems, and it does have the potential to become far more widespread than it currently is. It's on the doorstep of some of the hydropower schemes. It's not currently impacting the Waikato scheme, but it does present a threat. It forms in aquatic areas. In aquatic areas, it forms dense floating mats, and you can see here all that green material that's floating out over the water surface there is alligator weed. So very problematic in terrestrial situations. It forms monocultures, displacing, in horticulture, cropping, pasture, displacing other plant species that are present there. It's very difficult to control. So each node on a stem can produce a new plant. It easily fragments. It's producing through fragmentation. It doesn't produce viable seed in its introduced range. So mechanical clearance encourages its spread, livestock and those sorts of things. If you're fragmenting the plant up, there's potential for it to spread. Biological control agents are challenged by low temperature conditions where the weed still thrives. So the biological control agent, particularly the alligator weed flea beetle, is not as cold tolerant as the weed itself. So the potential distribution of alligator weed outside of the range where you're getting effective control from the flea beetle, it's going to spread further than that range. Large-scale, cost-effective management of alligator weed is reliant on herbicide applications, and different weed control outcomes have been achieved at different sites, but also in different countries as well. So some of this work was, we've presented this work previously. It was some work we did with Waikato Regional Council and Mississippi State University, where we looked at 6 different herbicide treatments in a lagoon at Tukawhai. And what we did there is we applied a range of treatments within that lagoon on 20 by 20 metre plots. So we had glyphosate, metsulfuron, triclopyr, and they were all single applications with repeated treatments at 4 weeks after treatment. And then we did combinations of those with a single treatment. And then we repeated these studies as well on a selection of those treatments in the USA. So these are some of the results that we found from that particular study at Tukawhai. So this is 12 months after treatment. So you can see the control up the top there on the right, the triclopyr retreatment and the glyphosate and metsulfuron treatment. If you have a look at the chart there, that was the only treatment that provided us longer term control over that 12-month period. All the other treatments were, at the same level as the control plots or greater at 12 months after treatment. And they didn't see this in the USA when they conducted trials, particularly with repeated applications of glyphosate and triclopyr, as well as glyphosate plus triclopyr. They saw 52 to 92% biomass reduction, and we just didn't see that in the trials that we had conducted in those field studies. So what could be happening there? And there's a range of reasons, but one thing that's come up is, are there differences in herbicide efficacy linked to the genetics of individual populations of alligator weed that are present? Okay, so this was some previous work that was done in the States, where they were looking at the genetics of alligator weed, and so they looked at 373 plants from across 90 sites. They were looking at the genetics at 3 chloroplast regions, and what they found was that there was high haplotype diversity. So haplotypes are a collection of genes that are inherited from a single parent, and it can be used to suggest that there has been multiple introductions within a region. So what was interesting about that as well was that they had high haplotype diversity, suggesting that there has been multiple introductions. And there on the right there, you can see there was 6 haplotypes, so AP1 to AP6, that they had identified. And 2 of those haplotypes corresponded to known biotypes. So a biotype's a group of organisms having the identical genetic makeup. And what's interesting about that, the 2 biotypes that were recognised were one with a broad stem and long, narrow leaves, and one with a narrow stem and short, broad leaves. And these biotypes are genetically distinct, they're different, and it's not a response to the environmental conditions where those biotypes had occurred. So what we did, in collaboration with a whole bunch of the regional councils and others across the country, and thank you very much for everyone that collected and sent the samples in. This sort of work can't happen without that. So we sampled from 16 sites across the country, as well as 13 sites in Australia, up the eastern seaboard of Australia. And at each site, 3 to 5 plants were sampled, and the samples included a leaf sample, which was removed, placed into sealable plastic bags and added desiccant gels, and then we dried the samples, and then they were sent for genetic analysis in the USA, using the same method as what they had done in that previous 2020 study. Okay, so some results of what we've found. Interestingly, we share 2 haplotypes, so AP2 and AP3, so the green and the blue dots on that screen we share with Australia. And this is the first study of its kind. So the sampling that we've done is still quite limited, so we could grow these data sets. But interestingly, we also have this third haplotype in New Zealand, AP5, which was not picked up in those Australian samples. So this is indicative of more than one introduction. So the presence of different haplotypes could explain the different weed control outcomes in the USA and New Zealand. There's a potential for this. The haplotypes that were present, so we're looking at the state of Mississippi here. The haplotypes that are present there, only one site that they sampled overlapped with the, so AP3, the green circle there, overlapped with what we have in New Zealand. So do we need different prescriptions for different types of alligator weed? Potentially. The plant response to weed control is more likely to be similar to what's occurred in Australia, except for that haplotype AP5, which are those purple points, which wasn't detected in those Australian samples, but is quite widespread in New Zealand. So the next steps for this work is to conduct an in-depth genetic study, so potentially grow those data sets. There was a couple of sites, one particularly in Bay of Plenty, that had mixed populations that were, so mixed haplotypes at that particular site. So sort of sorting through some of that would be useful. As well as conducting a herbicide efficacy study. So using our 3 genetically distinct populations, so selecting them, and I'll go into a bit of a study design, selecting those 3 and then running our herbicide treatments to see if they're responding to those control methods differently. So we're looking at starting that this year and sourcing plants next month, so we might be in contact with you again. In regards to the study design, we'll look at, so we'll be doing this in both in New Zealand and the USA. We'll be looking at the 2 haplotypes that are similar, that are present in both countries, so AP3 and AP5, and then we'll add the specific ones for our regions. And then we'll run these haplotypes against glyphosate alone, metsulfuron methyl, the combination treatment that was effective in the field, and then 2 other herbicides that are being used that are relatively new, not currently registered for use in New Zealand, fluprovoxafen benzyl and flumioxazin, and then a combination of those, and we'll see if it actually makes if that has an effect and if those haplotypes are responding differently. So, does alligator weed genetics influence weed control outcomes? Potentially, but verification is certainly required. And then a prescription for the alligator weed that you have in your region. And it's interesting looking at those maps and talking to those that are managing it and saying, okay, where's this site come from, and potentially being able to trace some of that using these genetic studies as well. So yeah, that's where we're currently at. I'd just like to acknowledge Denise Rendell for processing the samples at Earth Sciences, and then for everyone that was involved across the country for these collections. So yeah, thank you very much.
Awesome, thanks Daniel. Any questions for Daniel? Here.
Question, do the different haplotypes have morphological descriptions that we can use?
Well, 2 of the biotypes that were present in the US were separate. The 2 haplotypes corresponded with those 2 biotypes. So yeah, I think there's a bit more work to do there because, originally, we thought, oh, it's all vegetative reproduction and it's clonal. So those plants that look different that are in an aquatic and a terrestrial environment, are they the same are they responding the same, or is that a genetic difference now, or is it? Yeah, so I think that's what we've got to work through. In regards to herbicide response, I'm not sure how that's going. It hasn't been done before, but there has been work done previously on the biological control agents and responding differently to the different haplotypes. So, it's interesting space, and it's developing for other species as well. So, yep.
And do you think that they would respond differently in aquatic versus terrestrial situation?
I think so, for sure, particularly to the herbicides that we're using. Yep. Really interesting. Previously we were using a lot of triclopyr and glyphosate, metsulfuron as well, but I think there's some work there as well that we could do on whether that's optimising the rates of metsulfuron that are being used, because no one's looked at that to optimise, and if there's problems with residues and those sorts of things. I think also with translocation below the waterline, you seem to, with metsulfuron and triclopyr, it seems to remove the above water material very well, but you don't get the translocation below the waterline. So, yeah.
Any more questions? Have they done anything on the nuclear DNA?
No, look, I'm not a geneticist, if you can probably tell, but no, there hasn't been, as far as I'm aware. But the colleagues in the States are probably much further ahead than we are with that. There's a couple of papers out there, recent papers on that. So
you're saying that the haplotype differences were significantly different, like the haplotype and the control by the biocontrol was significantly different response?
Yeah, there was. Yes, correct. Yeah.
Someone up the back? Yeah, mate.
For those two herbicides that you listed, where you mentioned that weren't readily available, or just not available yet, could you maybe explain those again? Are they going to be released soon, or did you get a special approval to use this?
Sure. So we only use, so those products are not currently registered for use in New Zealand, but we will do efficacy testing in our laboratories before any of that happens. So generally, so that's the way that it works with sort of the development of the use of these products. So we start off small in lab environments. It has to be approved through the EPA for wide scale use, but for some of the experimental experimental lab trial stuff, we have a permit that we can do that under.
All right.
Yep.
Awesome. Thanks heaps, Daniel.
Cool.